Hello buddies, welcome back to the channel. This is Chandu ma'am and today we are going to discuss about the continuation of the chapter Sexual Reproduction in the Flowering Plants, and this is lecture number 4. In the previous classes, that is in the lecture number 3, we discussed about agents of the pollination and artificial hybridization. So in this chapter we are going to continue the concepts like double fertilization, post fertilization events and all. Most probably this will be the last class of this chapter. So what actually the double fertilization means? In examinations from this portion you may get much more questions for your theory basis also and for your MCQ point of view also. This is very important. See, when we talked about the pollen grain from the flower, in the flower from the anther, what happens after the maturation, pollen grains will be dispersed out. Okay, either it may be that pollination may be by wind, water, whatever the pollination agents may be, whatever the types may be. Once the pollen grain reach to the stigma, by the interaction of the chemical interaction in between the stigma and then pollen grain, there is a growth of pollen tube. This is the pollen tube, okay, and which is carrying the male gametophyte. So here this is the ovary, female gamete, where the female gametophyte is present. This is the female gametophyte. 1 2 3 4 5 6. Okay, now see here, one pollen grain is maturing and then it is moving towards the embryo sac. This is the embryo sac, entire thing. Okay, so in this embryo, we learnt about the embryo sac also. It is having how many? 7 celled, 8 nucleated. Yes, it is having 7 celled. 1 2 3 4 5 6 7 cells are there and 8 nucleus. 1 2 3, here at the center there will be 2 nuclei. 4 5 6 7 8. So there will be totally 8 nucleus and then 7 cells here. So what happens after entering one of these synergids? The pollen tube releases the 2 male gametes into the cytoplasm of the synergid. It is going to release its male gametes into one of the cytoplasm of the synergids. See here, there are 2 synergids, this one and this one. Among them, after that, let me draw here. Just imagine, here is the 2 synergids and there is a egg cell also, and there are antipodals, 3 antipodals. Okay, once the pollen tube is entered, that pollen tube will be entering to the one of the synergid, any one, either of this one or either of this one. By whom? With the help of filiform apparatus. Do you remember? We discussed about that. This is a finger-like projections which are present in the synergids. They will be attracting to the pollen tube. So that what happens? Pollen tube will enter to the synergids. After the entering, what happens? The male gametophytes will release. So after releasing here, one male gametophyte will move towards the egg. This is the egg actually. This one is the egg. Okay, one of the male gamete will fuse here and one more will be move towards the central nuclei where there is a 2 nucleus are there, polar nuclei, isn't it? So towards the egg cell and fuse with its nucleus to form a zygote. After releasing 2 male gametes from the pollen tube, one male gamete will fuse to the egg cell to form a zygote. Another male gamete will move towards the polar nuclei. That's what it is called, what? Double fertilization. Here fertilization occurs 2 times. This one is syngamy. Syngamy means syncage of the gametes. So one egg cell is the female one and then male gamete is came, so they are going to be fused. Due to that, what happens? There is a formation of the zygote. This is one fusion. One more male gamete is going to move towards the polar nuclei. These two are the polar nuclei, isn't it? So once it is joining here, we will be finding primary endosperm nucleus, that is PEN, pen. Okay, yes. So this is the one fertilization, and then this one is two fertilizations. That's what it is called as double fertilization. Okay, the other male gamete, one is moved here and it is fused, that is called as syngamy. One more is moved towards the polar nuclei located in the central cell and fuses with them to produce what? Triploid. Just keep in mind, this one is triploid, primary endosperm nucleus, that is PEN. Triploid means what? Here the number of nucleus is n, 3n, that's what it is, triploid. And here egg, after the fusion of this female and then male gamete, it is giving you diploid. Zygote is diploid, but primary endosperm nucleus is triploid. As it involves the fusion of 3 nuclei, it is also called what? Triple fusion. Which one? This one. Okay, this one is also called as what? Triple fusion, because there is a fusion of 1, 2, 3 male gametes, isn't it? That's what it is also called as what? Triple fusion. Next, since two types of fusion, syngamy and then triple fusion, takes place in the embryo sac, this one is called what? Double fertilization. Why it is called double fertilization? Because inside the embryo sac there will be a fusion of two types of things are going to take place, that is fusion is going to take place, syngamy and then triple fusion. That's what it is called what? Double fertilization. Next, what happened? This one, this central cell after triple fusion becomes the primary endosperm cell. Wait, I will take a slide here so that I can explain you. Let me explain you here. I will draw. See what did I say? Just imagine this is the stigma and this one is the style. Stigma, style and ovary. For your convenience, I will take only one ovule. Okay, this one is the ovule and it is having the embryo sac. Okay, I will zoom this embryo sac here. This is the embryo sac. Let me take synergids here for convenience. And this is the egg cell. This one is egg. Okay, and here are the filiform apparatus. And opposite to them there will be antipodal cells, 3 antipodal cells, and centrally this is overall thing, this overall cell is central cell and it is having 2 nucleus, 1 and then 2 nucleus, which is called as polar nuclei. Polar nuclei, and these are the antipodals. Okay, I am zooming this portion, only this portion. Okay, see, once pollen grain is attracted to the stigma and then there is a coordination. If everything goes smooth, the stigma will allow this pollen tube to enter. So pollen tube will grow along with the style and then it reaches to the micropyle end of the ovule. Once it reaches to the micropyle end of the ovule, whatever the pollen tube is there, that is going to be immersed inside. Due to what? Due to the help of the filiform apparatus. These filiform apparatus will guide the pollen tube, whatever the pollen tube is entering. So I will show you the pollen tube here. So pollen tube, now it is having, let me take another color so that it will be. So it is having 2 male gametes. This one is one male gamete and this one is the another male gamete. 2 male gametes are there. So this tube, pollen tube, grow towards the one of the synergid. towards the one of the synergid, either one, this one or else this one. After reaching the synergids, it will burst off, the pollen tube will be burst off, and then two male gametes are released. After releasing, what happens? This is synergid, this is egg cell. Just imagine pollen tube is entered and then it is burst off. After bursting, what happens? Whatever the male gametes are there, they are going to be released. Among these two male gametes, one is going to be fused to the egg cell by the process called as syngamy, isn't it? To form what? This is female gamete and then this one is the male gamete. They both fuse to give what? Zygote. So here the zygote will be 2n in number because 2 nucleus, there will be two forms of the DNA. So this is what actually. And what happens to the another one? Another male gamete. Where is another male gamete? See here, there is antipodal cells, and there will be this one is a central cell which is empty now, and I am drawing now with polar nuclei which is having two nucleus. Okay, polar nuclei is having what? Two nuclei. So now what happens? One of the male gamete is fused with the egg cell, you know that, to form what? Zygote. That process is said to be what? Syngamy. One more remained male gamete is there. Now it is going to move towards the central cell, and then where the polar nuclei is present, and it is going to fuse with that to form a 3n number. 3n, which is said to be what? Triple fusion, which is called what? Triple fusion. Why? Because 3 nucleus are going to be fused there. This is called syngamy because two gametes are going to be synchronized. And whereas in the angiosperms, this type and then this type of fertilization is going to take place. That's what it is called, what? Double fertilization. I hope everybody is clear about this. Next, what happens? This zygote is going to form the embryo, and then whatever this pen, PEN, I can say, primary endosperm nucleus, PEN. Okay. Triple fusion result is PEN, and then double fusion, this one syngamy result is zygote. Next. So when we talk about the post-fertilization structure and events, post-fertilization actually has 4 events. One is endosperm development, embryo development, ovule which is going to form into the form of the seeds, and then ovary into the form of fruit. How actually these are going to be happen? One by one we are going to discuss about these all things. So firstly, what happens? Before the embryo develop, there will be some amount of endosperm is going to be developed first. So first we will talk about the endosperm development. See here, these synergids are already degrading, and then zygote is there. Zygote is a fusion of the male and then female gamete, isn't it? And this one is the PEC, that is primary endosperm nuclei, or sorry, cell, and PEN is primary endosperm nucleus. PEN, PEC. This is the cell. Inside this cell there is a pen which is 3n in number due to the triple fusion, and then antipodal cells will be degenerated. Antipodal cells and then synergids after fertilization, they are going to be degenerated. The primary endosperm cell divide rapidly and form a triple endosperm nucleus. Okay, this one. Which one? This. That is PEC. PEC will divide rapidly to form triploid endosperm tissue. Just imagine this is PEC, only this cell. Just now focus in this cell. Okay, whatever the blue outline I said, no, that one. Eggs, this one zygote is going to form a embryo. Don't worry about that. Now we are going to focus this one, this area, only this portion. This endosperm, only now it is having only one cell, isn't it? PEC, this one is PEC. Due to the many multiple divisions, what happens, it is going to form a tissue. That is one is called as triploid endosperm tissue. 1 2 3. After multiple of the divisions, it is going to form a triploid endosperm tissue. This is the endosperm tissue. Endosperm cells are filled with reserve food materials and are used for the nourishment of the developing embryo. Whenever this embryo is going to be developing, no, to that embryo there must be a nutrition, isn't it? That nutrition is supplying by whom? By the endosperm tissue. See here, this pen is there, no? What happens? The pen undergoes successive nuclear division to give free nuclei. There will be only division of nucleus. 1 nuclei, 2 nuclei, 3 nuclei. Many nucleus will be divided, but not there will be no formation of cell wall after immediate formation of the nucleus. So due to what happened, due to that what happens, there will be a endosperm development which will be showing you free nuclear endosperm. So you can see in the coconut, isn't it? The endosperm undergoes successive nuclear division to give free nuclei. This stage of endosperm development is called as what? Free nuclear endosperm. Only nucleus is going to be divided. In PEC, that is primary endosperm cell, there is a pen, primary endosperm nucleus. That nucleus is only dividing. There is no formation after dividing of the nucleus. There is no formation of the cell wall. So the endosperm becomes cellular due to cell wall formation later on. Okay, the coconut water from the tender coconut that you are very familiar with, that is nothing but the free nuclear endosperm. Whatever we are going to drink the water, no, that is coconut water. That one is nothing but what? Free nuclear endosperm. Okay, made up of what? Thousands of nucleus, which is 3n in number, isn't it? Yes. Whatever this one white kernel is there, no, it is cellular endosperm. Got it? Whatever the thing will be there here, white one, watery like structure, that is free nuclear endosperm, and the white kernel out which is harder one, that is called what? Cellular endosperm. See here, this one is the tender coconut, we can say. This one is the seed coat, and this is the water, and this one is the white kernel. Okay, this is the liquid one is coconut milk, I can say, I can call it as, and it is liquid endosperm, that is free nuclear endosperm. Whereas this one white one, whatever the solid endosperm is there, which one will be cellular endosperm. Next, after the endosperm formation, what happens? There will be a development in the embryo, that is the next stage. So here in the embryo development, what happens? Embryo develops at the micropyle end of the embryo sac, where the zygote is situated. When we talked about the embryo sac, synergids are going to be degraded, isn't it? There is only presence of zygote. This zygote is at the micropylar end. So this zygote will develop at the micropylar end, and it is going to be to form what? Embryo. Embryo develops at micropylar end of the embryo sac, where the zygote is situated. Zygote divide only after certain amount of endosperm is formed. This gives nutrition to the developing embryo, though the seeds differ greatly, the early stage of embryo development is same. See here, this is the nucleus of the zygote and this one is the vacuole. This is entire embryo sac. Okay, what happens here, the nucleus of the zygote is going to be divided to form a two-celled proembryo. Okay, and frequently there will be a globule, heart shape, and then mature embryo, which is already given your NCERT. That process itself is said to be what? Embryogenesis, the formation of the embryo. See here what happens, there is the one cell which is said to be a zygote, which is going to divide to give you two cells. One is apical cell and then one more is basal cell. Basal cell is going to form the suspensor in the future, and then apical cell is going to be form the embryo properly. Okay na? So this basal cell again divided into many forms, and in the stack of these basal cells, first one is called as what? Hypophysis, and remaining things are called as suspensor. And here this is the 16-celled embryo. After that what happens? Globular embryo, because it is round in structure. Next there comes the heart shape. Why this heart shape? Because there is a small projection inwards, which leads to formation of the cotyledons. So embryo development here will be firstly proembryo, next globular, next mature embryo. Next, this thing, whatever the embryo is going to be formed up to the heart shape, this is same for monocots and then dicots. Okay, see, this is the example of the dicotyledonous embryo, and then this one is example for the monocotyledonous embryo. This is the embryo and here is the embryo. But why it is called as monocot and then dicot? Monocot, it is having only one cotyledon. Dicot, it is having two cotyledons. Okay, see here, one, this one is one, and then this one is two cotyledons, and here only one cotyledon. Which one? This cotyledon. Okay, yes, it has an embryonal axis and two cotyledons. Which one? Dicotyledonous embryo. It has the embryonal axis. This is the embryonal axis, I can say. The portion of embryonal axis above the level of cotyledon is epicotyle. Okay, above the level of the cotyledon. Which one is called was epicotyle, which terminates with the plumule. See, these are the cotyledons, above which is said to be epicotyle, and then below is said to be what? Hypocotyle. Hypocotyle region contains radicle which leads to the root, whereas epicotyle will be having the plumule, which is a stem tip. This is actually the plumule, which is having what? Stem tip. The cylindrical portions below the level of cotyledon is hypocotyle, that terminates with the radicle, that is root tip. See, this is the portion level of cotyledon. If you imagine, above that which is said to be what? Epicotyle. Below that which is said to be what? Hypocotyle. Hypocotyle contains radicle and then root cap, which is going to be differentiated in future in the form of root, whereas epicotyle contains plumule, which is the stem tip. In future what happens? This portion only will give you stem. Here the root tip is covered with the root cap. Whereas in the monocotyle, this is about the dicots, and this is when we talk about the monocotyledons, they possess only one cotyledon. Cotyledon of the grass family is called as scutellum. Here, see, scutellum, this is given, actually blurred image is there. Give me a minute. Okay, so let me explain you in detail the dicotyledonous embryo. So this is the clear image of the dicotyledonous embryo. Sorry for that, the image is not that much clear. See, above the, if we take, this is the level of the cotyledon, above the level of the cotyledon, which is called what? Epicotyle, isn't it? Yes, epicotyle, and below that which is said to be what? Hypocotyle. Epicotyle contains plumule. Plumule means what? Here there will be stem cells. Many of the thousands, millions of the many cells, small tiny cells will be there, which will be giving you in future what? Stem. Okay, that one is also called a stem tip. Whereas in the hypocotyle we will be having, this one is the radicle, and then this one is the root cap. This one we used to see in the dicotyledons because it is having two cotyledons. This one is the one and then this one is the two. But whereas in the monocots it is having only one cotyledon, which is called as what? Scutellum. So next is about the monocotyledons. This is all about the dicot. We will talk with the monocotyledonous embryo. Here what happens? They possess only one cotyledon. The cotyledon of the grass family is called as what? Scutellum. This is the cotyledon of the grass family, which is said to be what? Scutellum. It is situated laterally to the embryonal axis. It is situated to the lateral side. This is the lateral. This is embryonal axis. This is the embryonal axis. Okay, which is present lateral. Lateral means side by. Okay, scutellum is lateral side of the embryonal axis. At its lower end, embryonal axis has a radicle and a root cap enclosed in coleorhiza, that is an undifferentiated sheath, whereas the portion of the embryonal axis above the level of the scutellum is called what? Epicotyle, and it has a shoot apex and a few leaf primordia enclosed in the coleoptile. Okay, now a hollow foliar structure. See, let me explain what actually this is. See, this is the embryonal axis, whatever you are going to see. This is the level of the cotyledon. Above that, which is having the shoot apex. This shoot apex is covered with the shoot sheath called as coleoptile, but we didn't have any kind of sheath here in the dicot, but we have in the monocot. The sheath which covers the shoot apex, which is called as coleoptile, and then below this axis, embryonal axis, there is a presence of the radicle which is covering root cap. But these two are covered by one more sheath which is called as what? Coleorhiza. Coleorhiza is for the root, coleoptile is for the stem. Keep in mind, many of the times they asked and then students will get confused here. Coleorhiza, rhiza means the word itself says the root. Coleoptile, which is going to be a covering sheath, the protective sheath for the shoot apex. Okay, this is about the monocot and then dicotyledonous embryo. Now let's move towards the next portion, that is this from seed from ovule. How actually ovule gives rise to seed? In the dicotyledonous seed, and then this is the dicot seed, and then this one is the monocot seed. This is only one, there is only one cotyledon. This is the cotyledon. Okay, inside the cotyledon there is a embryo. So overall this thing is embryo, and here there is a two cotyledons. This one is one and then this one is the two. Let me. Seed is the fertilized ovule formed inside the fruit. Seed is nothing but what? It is a fertilized ovule. It is the final product of sexual reproduction. Which one is the final product? The seed is the final product. It consists of seed coat, cotyledons and embryo axis. Yes, whenever we are going to discuss any of the things, see, this is the embryonal axis and these are the cotyledons and this one is the seed coat. Outer hard structure, whatever is you are going to see now, that one is the seed coat. The cotyledons are simple, generally thick and then swollen due to the storage food material. This one, storage food material. Okay na? Again here there will be two types, that is albuminous and non-albuminous. What actually albuminous means? Endosperm. If endosperm is present in the mature seed, then it is called as what? Albuminous. If it is not there, then it is called as non-albuminous. What happens to this? Why there is a two kind? See, if the developing seed, in the mature seed, developing embryo consumes all the endosperm, then that comes under the non-albuminous. If there is a leftover albuminous, then they comes under the albuminous. Okay. Here, in albuminous endospermic seeds, what happens? There is a presence of mature seed, endosperm is present, whereas non-albuminous there is will be the absent. What about the food storage? Here endosperm is the food storage area, and then here the cotyledons will be there. Embryo, here it will be very small, and here there will be a large and then well developed endosperm. Why? Because it is consuming all the endosperm. That's what it is, large and then well developed. Food used during development, not fully used, because of that only it is called as albuminous, because the presence of the albumin will be there, and it is fully used, because of that everything is khali, so that embryo is fully consumed all the endosperm. Examples for this: wheat, rice, maize, castor and then coconut. Example for the non-albuminous: pea, bean, gram and then groundnut. Okay, here I gave some of the diagrams. This is the bean, which is the non-albuminous, and this is the corn, okay, which is the off monocot, and here this is the castor, which is albuminous. Wheat is also albuminous, and then maize is also albuminous. Occasionally in some seeds what happen, there will be a remnant of the nucleus. Remnant means leftover of the nucleus will be there, which is called as what? Perisperm. Persistence means present. Okay, now if in a seed there is a nucleus is present, means then it is called what? Perisperm. It is already asked many of the time, and even for the theory there may be chances of asking for the 1 marks. Integuments of the ovules harden as tough protective seed coat. It has a small pore through which oxygen, water enter into the seed during the germination. There will be a small pore, which is, if you usually see any kind of this seed, there will be a small portion. Let me draw a diagram of one seed. This is not hard. I'm drawing the seed. Okay, this, above which there will be a small portion which is called as micropyle, where the entry of the oxygen and then water is going to be takes place. What happens when there is an entry of the oxygen and then water? This embryo starts growth. See, here it is. Here is the seed, isn't it? There is a micropyle end. From this, if the water and oxygen is absorbed, then there will be a germination of the seed. As the seed matures, it becomes dry by reducing water content 10 to 15% moisture by the mass. Okay, it will be reducing that much. But the metabolic activity of the embryo slow down. It may enter the state of what? Inactive stage, that is the dormancy period. What is actually the dormancy? In simple words, you can say that it is a resting period, where the metabolic activity will be very slow down, but it is not dead. Okay, again under favorable condition, what happens? If you give, you can store the seeds in the at our home, isn't it? We can store them, because that seeds will be under the dormancy period. If you expose those seeds to the water, what happens? They are start germinating after some time, isn't it? So when undergoes favorable condition, when they get the moisture, oxygen and then temperature, they germinate. They start to germinate, to form to give roots and then leaf, etc, etc, etc. So what are the advantages of the seeds? Due to the dormancy of the seeds, we have much more advantages, isn't it? Since the pollination and then fertilization are the independent of the water, because for the pollination and then fertilization water is independent. Now, there is no need of the water, compulsory is not there, isn't it? As like in the bryophytes and then algae, here we are talking about the angiosperms. Seed formation is more dependable, better adaptive strategies for the disposal to new habitat. It helps the species to colonize in other areas. What it means, what he is going to explain here. See here, as we are the pollination and then fertilization are the independent of the water, seed formation is more dependable of what, for the adaptive stages, like what, just imagine when you eat mango, you will eat all the fruit, but you will remain with the seed. Will you leave that seed in wherever the mango tree is given? No, you will throw it somewhere else, so that what happens, that mango is colonizing somewhere else, means there is a species aeration, we used to call that one. They have food reserve material, so that the young seedlings are nourished until they are capable of photosynthesis. There will be a food stage. Due to the food storage, due to the very much of the nutrition, we used to eat the cereals and then millets, etc seeds. Okay, next, parts of the seed. This is the seed coat, this is the embryo, and then this is the stored food. The hard seed coat protects the young embryo. This hard seed coat protects what? Embryo. Being product of sexual reproduction, they generate new genetic combination and then variation. Due to the production of the sexual reproduction, it is having what? New genetic recombinations. Dehydration and then dormancy of mature seeds are crucial for the storage, for the strong of seeds. It can be used as a food throughout the year, and also to raise crop in the next session. You can store this one, either you can store for the future purpose, or else you can sow them for so that you can get a plant. Next, viability of this seed. What is actually viability? That is the capability. Okay, now that the seeds capability may be for 2 days, months, years, many years, like that. Okay, in few species what happens, the seeds lose viability within a few months they can lose. Seeds of many species live for several years also. So for that, NCERT given many of the examples. Just keep in mind whatever they here highlighted. In some seeds can remain alive for hundreds of years. The oldest is that of lupin. The oldest one is the lupin, that is Lupinus arcticus. Here this is the lupin one. From Arctic tundra, the seed germinated and flowered after an estimated record of 10,000 years of the dormancy. How much year of dormancy? 10,000 years of the dormancy. Next, 2 years old viable seed is date palm, which is Phoenix dactylifera, discovered during the archaeological excavity of the King Herod's place near the Dead Sea. So 2000 year viability is for the date palm, and 100 years of the viability for the lupin. Now, formation of the fruit. Fruit is going to form from the ovary. So the ovary develops into what? Fruit. Transformation of ovules into seeds and then ovary into fruit produce simultaneously. See, just imagine this is the ovary and then this is the ovule. This is what? Ovule. This ovary develops into fruit and then this ovule develop into seed. That's what, inside the fruit we will be having this seed. Okay, yes. The wall of ovary develops into pericarp, which is called as wall of the fruit. The fruit may be fleshy, or else they may be dry, like dry fruits. You can also give examples. If fleshy, example guava, orange, mango, these all are fleshy fruits. Guava, orange, mango, this one is the guava, and these are the dry fruits. This is the groundnut. What actually? Okay, this overall thing is fruit. Groundnut is this seed. Will you find any fleshy thing here? No, that's what it is called. What? Ground dry seed. Sorry, dry fruit. Next, again there are two types in the seeds, that is true fruit and then false fruit. This is depend upon the presence of the fleshiness. Okay, if it is present, pericarp is present in the form of fleshy, that is called as fleshy fruit. If it is dried, then it is called dry. But this one is like true fruit means it is the development of fruit from the ovary. Okay, if the fruit is truly the production of the sexual reproduction, they come from the ovary. If not, in this, thalamus is also contributed to the formation of the fruit, then it is called what? False fruit. Thalamus means what? Thalamus means it is the end portion of, see, this is the stalk of the flower. There will be a bulged portion which is called as thalamus. On this there will be a formation of the petals, stigma, style, and then ovary, and then anther, those all things. If this thalamus is involving in the formation of the fruit, then it is called what? False fruit. If it is not, it is directly ovary is forming the fruit, then it is called true fruit. Examples are given here. This is the apple and this is the strawberry, and this one is the cashew. Apple, strawberry, and then cashews are examples for false fruit, because whatever you are going to eat this portion in the apple, no, that is actually nothing but the thalamus. Inside whatever we are going to usually we remove, no, this one is actually the fruit. This one is the wall and this one, these are the seeds. Okay, yes. Next, in some species fruits develop without fertilization. If the fruit is developed without fertilization, then that kind of fruit is said to be what? Parthenocarpic fruits. Parthenocarpic, usually they will be seedless. Best example, banana, pineapple, watermelon, etc, etc, etc. If the fruit is not having the seeds, then they will be parthenocarpic fruit. Parthenocarpic fruit means fruit develops without fertilization. Parthenocarpic can be induced through the application of the growth hormones. You can induce them by applying the growth hormones. Next, let's talk about the apomixis and then polyembryony. They are also very important, and this is the extra shortage we can say. Apomixis is the production of seeds without fertilization. Okay, production of fruit without fertilization is this one, parthenocarpic. Production of seeds without fertilization is called what? Apomixis. Okay, like in some of the Asteraceae and then grass family, these all are the Asteraceae family members only. Here what happens, there will be formation of seeds, but there won't be a fertilization. Okay, that's what the apomictic things are said to be. What they are the forms of sexual reproduction, but mimics a sexual reproduction. Okay, see, here are the seeds. This is the seed. Seeds without forming fertilization is called apomixis. Fruit without forming fertilization is called as parthenocarpic. Don't get confused here. Next, how the development of the apomictic seeds is going to take place. In some species, diploid egg cell is formed without reduction division and develop into the many of the embryos. Then it is without fertilization. Okay, there will be formation of what? Embryo without fertilization. In few of the species we can say. In many species, some nuclear cells surrounding the embryo sac divide, protrude into the embryo sac to form embryos. Thus, each ovule contains many embryos. What happens in few of the species, if I say there is a nucellus, okay, and here is embryo. Whatever the nucellus cells are there, if they are protrude into the embryo sac, what happens? There will be a formation of multiple embryos, which condition itself is called what? Polyembryony. Poly means many, embryos means embryos. Occurrence of the more than one embryo in a seed is called what? Polyembryony. Due to the polyembryony, there will be a, see here, this is only one seed is sowed, but you got two plants, isn't it? Here also same thing, one seed will give you two because of the presence of more embryos there. Next, importance of apomixis in hybrid seed industry. This is a big industry, I can say. Nowadays we are going to hybridize many of the things, whatever the characteristic we are going to want, no, means in a breed whatever kind of characteristics are wanted, we can go with the apomixis. If seeds from hybrid are sown, the plant in the progeny will segregate and lose hybrid characteristic, isn't it? Yes. Production of hybrid seed is very costly. Because of that, what happens, hybrid seeds are also very expensive. Because of the hybrid seeds are very expensive because production is very costly. If the hybrid seeds are made into apomictic, what actually hybrid seed means, if two characteristics are hybridized in a one, then it is called what? Hybrid seed. Okay, this is the hybrid seed, but forming of this is very very costly. Because of that, we can, what we can do, we can optimistic, okay, we can go with the process of apomixis, so that there is will be a no fertilization only, isn't it? But there will be a formation of the seed. Okay, there is no segregation of the character in the hybrid progeny, so farmers can keep on using hybrid seeds to the raise new crops. To raise new crops, there will be a very useful this one, because they will be having the progeny which is of the hybrid progeny, in the sense the important characteristic which we wanted to be sowed in that plant. Okay, this is what actually the use of the apomictic we can use. So this is all for today, and this is the end of the chapter, that is sexual reproduction in the flowering plants. For your NEET point of view, you may expect 2 to 3 questions from this chapter, so please read this chapter line by line. If you compare my notes to the NCERT, almost all the same lines I have taken here, because as I said, line by line NCERT means purely it will be of line by line, because in the examination he will be asking directly, he will be asking indirectly also. That's what you must be very well prepared for the examination. So this is all for today. Thank you very much.